Thank you so much, Claire. It's an honor and a pleasure to come here and give a talk. When I was a student about 55 years ago, I used to come to the Philosophical Society talks. So, this is now 55 years is a long time. A great pleasure. So it's also a particular honor to give a talk because the Philosophical Society and the Cambridge scientific community in general have been very important in the development of crystallography and structural biology over the years. And so I thought I'd start by showing you a few panels that are taken from a paper that was presented at the Philosophical Society in, you can see, November 1912, presented by J.J. Thomson on behalf of his student, Lawrence Bragg, who had learned about the X-ray diffraction pattern that Friedrich and Knipping took with Laue in Munich earlier in 1912, and then worked out how these spots could be derived from the diffraction of X-rays by the crystal structure, and introduced this Bragg's law, which is 2d sine theta equals lambda, although in those days they used a different definition from theta that we now use. And so this, in the hands of Bragg and his father, let them determine an atomic structure for this particular crystal, zinc sulfide, but also sodium chloride, diamond, and lots of other structures. And by now, so this is now over 100 years later, there's now over 1 million structures of crystals in the Cambridge Crystallographic Data Center right next door here that was initiated by Olga Kennard a while ago. And then in parallel with what you might call smaller molecule crystal structures, structural biology developed also a lot in Cambridge, although elsewhere as well. And this is a sort of mini timeline for the first 50 years after the discovery of X-rays and the electrons and Bragg's law. But there was Bill Astbury and J.D. Bernal were here in the Cavendish, and they went off to Leeds and to Birkbeck to develop fiber diffraction and crystallographic diffraction in what later on Don Caspar, who coined this name structural biology, said he thought was the beginning of structural biology. And then there are lots of other things, crystals obtained, structures, and I'll look at a few of these things, giving you the first 50 years. So this was the beginnings of protein crystallography in Cambridge, in the Cavendish lab, by Max Perutz and John Kendrew. And this was pictures of sperm whale myoglobin, and these were the charcoal sketches that Lawrence Bragg, who was the Cavendish director at that point, that was his hobby, drew in 1964, soon after they got structures for hemoglobin and myoglobin. And the first structure of myoglobin was this, the low resolution structure of myoglobin from X-ray diffraction patterns like this one, was the beginning of the very first structure of the first protein done by crystallography. And with a bit more effort over the next 2 years, between 1957 and 1960, they got a high-resolution structure with all the atoms of myoglobin. This was a painting that Irvin Geiss painted and is now hanging in the Howard Hughes. They bought it recently in near Washington. So that was the beginning of protein crystallography. One structure in 1960, another one in 1964, lysozyme, and then it carried on a few per year. And then about 30 years later, in the 1990s, by then there were 3,000 or 4,000 structures. And one of the interesting things I wanted to pass on is Max Perutz told me at coffee one day at the MRC lab, he said, "I think there are too many crystallographers now, given the great success of the method." And you can see the productivity by year. They were doing 20 or 30 structures per annum. And then suddenly, with freezing of crystals to give you a better lifetime and the new third-generation synchrotrons, suddenly the productivity increased by a factor of 30 or 40 until there were now several hundred new structures per year. And that was the background against which Max had made this observation. And so I asked him, I said, well, it seems to me the structural biology is just becoming very productive. What makes you think there are too many crystallographers? By which he meant that there weren't enough problems, enough structures to work on, and the number of people doing it, so they were competing with one another. And he said that quite often he opened Nature every Thursday or Friday, and he would find 2 articles describing the same structure by completely different groups. And I said, well, maybe that's very rare. And he quoted, this was the one he quoted, was NF-kappa B, which is a transcription factor that controls expression in immunology, infections, and so on. And these 2 groups, one of them is at Harvard, one of them is at Yale, and both of them are funded by the same Howard Hughes Medical Institute. So, he was right to notice it, but most of us disbelieved him until a year later, I opened Nature another time, and it turns out then there's 3 articles describing the same structure. This was Cytomegalovirus protease, which would be a drug target to prevent people getting infected by Cytomegalovirus. So at that point, I went along to some of the slightly younger crystallographers. I said, Max might be right from a year ago. Maybe it's true that there aren't enough structures, or too many. And Phil Evans said that I didn't know half of it, because it turned out there was another paper published 2 weeks earlier on the same structure by a different pharmaceutical drug company. And then there were 3 other groups that Phil Evans knew were working on it. So there were 7 groups all working on the same structure. So that was a little bit about the background to the growth in X-ray crystallography. So why then? So one other thing. So I thought it would be good to jump to the present time, and now we have this coronavirus infection. So over the last year, or year and a half, 2 years, it turns out, and I heard this from Janet Thornton, in the protein database where people deposit all their structures, in 1 year there were over 4,000 new structures of these 3 proteins that are the key drug targets for trying to ameliorate the coronavirus infections. The spike protein on the outside of the virus, the polymerase, and the protease that cuts up the proteins so that they can do their job. And I just picked out one paper. This is from a group in Tsinghua that was published a year or so ago. And this is an image. That protein there is one of the proteins, just one of hundreds of thousands of structures that are now known. And this is the main protease in the coronavirus that cleaves up a polyprotein so that 10 or 11 different proteins can do their job and allow the virus to infect it. And what you have here is the protease that cleaves the protein and a drug bound to it. And this is Paxlovid, which is the new Pfizer drug, which I expect they will be giving to the Queen at the moment because it's very effective in cutting down the consequences of infection. And that's its structure. So it's a small molecule binding to a protein and inhibiting the activity. And so now we have hundreds of thousands of structures. Let me go again. Yeah, so given that background, and with 180,000 sets of coordinates, mostly determined by X-ray crystallography, why do we need electron cryomicroscopy? And one of the things was I was working back in the 70s, trying to work on a membrane protein, Bacteriodopsin. We couldn't get crystals. So you were completely stuck, yet it was a very interesting structure. And then there were many other proteins, protein complexes, key parts in the molecules of life. Many of them were just too big to crystallize, or if they crystallized, too big to diffract in those days. They were too flexible, so if you got a crystal, it didn't diffract to high resolution. It was heterogeneous, so it was a mixture of different states. It was unstable, so you purified it, but an hour later it was gone, and so you didn't have time to crystallize it. Or you could get tiny amounts of material, and you need enough to make crystals. So there were lots of reasons why the available methods, crystallography or NMR, weren't really quite as good as they should be. And then against this background, I think a lot of people knew that electron microscopy in general had a lot of potential. And John Kendrew, who was the lead author on all, and the leader of that work on the myoglobin that I showed you earlier, he gave 6 half-hour lectures on BBC TV in the 60s, and in that showed lots of pictures, and there's a book published. And if you look in the book, there are about 15 or so X-ray diffraction patterns, which was his main topic, his main technique that he used. But also in that book, there were 20 or 25 pictures with the electron microscope, but they were low resolution with stains and things like that. So I think John Kendrew and many other people realized that electron microscopy in general had a lot of potential for developing in power. But that was the reason for being interested in it. So the first time I heard a little bit about that was when in the MRC lab, Aaron Klug, working with David Erosia and Tony Crowther, came up with 3D models from these 2D images. But again, these were images of biological structures, tomato bushy stunt virus and T4 bacteriophages that were stained with heavy metal stains, uranyl acetate, potassium phosphotungstate. But from images like this, because of the symmetry in the helix and the symmetry in the icosahedra, you could end up with a 3D model of it. And that was the work that was done initiating 3D structures from 2D images in electron microscopy. But my own, let's say, transition from being an X-ray crystallographer to being initially an electron crystallographer came from meeting Nigel Unwin, or listening to a talk that Nigel gave in 1973 in the annual seminar series in the lab. And Nigel was working on viruses, but he was thinking about how the electron microscope images might contain information about the protein structure, not just about the stain that was surrounding it or the stain that was covering it. And so we worked for a year or two together. This is us looking a bit younger in the earlier years, with diffraction patterns and images of these specialized patches in the membrane of Halobacterium salinarium, used to be called Halobacterium holobium. And this is a freeze fracture picture taken by Walter Stachinius in San Francisco. He discovered these specialized patches. They're a purple color. They're 2D crystals. There's about 100 molecules in each direction, so 10,000 unit cells, diffraction patterns like this. And we were able to get, at room temperature, not cryo microscopy in 1975, this model with 7 alpha helical stretches of the polypeptide, similar to the alpha helices that you saw in that myoglobin model earlier on. And then we carried on, and after about 15 years, we got a higher resolution model by going from 7 angstrom resolution to 3. And it's only a factor of 2 or maybe 10 times in 3D data, but it was much harder to get the information and needed the beginnings of cryomicroscopy, but using electron diffraction. And so that was how I got in, thanks to Nigel, made the transition from being an X-ray crystallographer to being an electron crystallographer. But the real power and the real, let's say, the opening of the curtains to show how cryoEM should happen and should allow you to solve some of these problems that were outstanding came from the work that Jacques Dubochet did at the EMBL in the early 1980s. And he was recruited to EMBL by John Kendrew, who I mentioned did have a great enthusiasm and a great anticipation that electron microscopy is going to be important. So Jacques was recruited from Switzerland, where he'd been a PhD student, to be a group leader at the EMBL to develop ways of freezing thin films of molecules so that you could do electron microscopy. And he also developed a liquid helium temperature, very cold electron microscope. And the key method, which we still use now, it's now 40 years later, is that you take a little EM grid, this is 3 millimeter grid, put a little drop, 2 or 3 microliters of your solution, which could have viruses, molecules in it, and then blot it and plunge it into liquid ethane at the temperature of liquid nitrogen. And he had this gravity-fed and elastic band-powered plunge freezing apparatus. And now you get just a computer-controlled version of this, but it's exactly the same thing. And then one of the first specimens that Jacques took, he took pictures of lots of things, and there's a really good review with maybe 20, 30 different examples, not very high resolution initially, but one of the first pictures he took was this one of Adenovirus, which was published in Nature in 1984. And you can see most of these adenoviruses are about 1,000 angstroms in diameter. Most of them are intact, but a few of them are either broken open or completely destroyed by being blotted by this air-water interface in the blotting. And it took them about 5 or 6 years to go from images like that to a 3D model like that, following along the lines that I mentioned Tony Crowther and Aaron Klug had done with negatively stained images of virus. But this is now unstained viruses in a thin film of ice. So what you're looking at here are the atoms that are of the protein, not the atoms of the stain. And then it took quite a long time to go from this, which is a low resolution, a sort of blobby model of the virus, to get images like this. So this is 1991, this is 2010. So 20 years later, better images from better microscopes with better vacuums, brighter sources, and so on. And this is taken by Hong Cho's group at UCLA. And when they processed those, they got a much more detailed picture of the adenovirus than they were able to get in 1990. So 10 times the resolution, 1,000 times as much data. And looking at one of the subunits, you see the beta strands, side chains of the protein. So that was technical improvements in the electron microscope that allowed you to do cryo-electron microscopy, but at higher resolution than before. And so about 20 years ago, Tim Baker and I wrote a review specially for the structural biology community, the X-ray crystallographers, in the International Tables for Crystallography Volume F, which had about 90 different chapters. And there are only 2 of them that involved electrons, one on electron diffraction, which was written by Hua Chu, and one of them on electron microscope imaging, which Tim and I wrote, picking out examples in the year 2000 of specimens that were amenable to this type of electron cryomicroscopy. This was 70S ribosome from Joachim Frank's group at 11 angstrom resolution, still blobby. This was hepatitis B virus cores done by Bedina Potcher, who was a postdoc at the LMB, and that was the first time that a single particle, and there was not a crystal, obtained a resolution that was beyond 10 angstroms. And then this was thin filaments of actin decorated with myosin subunits from Ron Milligan's group, now at Scripps. And blue is actin, green is tropomyosin, and then the myosin that attaches to the actin and pulls the filaments along to cause muscle to contract are these pink, yellow, and orange subunits. And then this was another 2-dimensional crystal, like the one that I showed you with Bacteriodopsin, that Werner Kuhlbrandt, who was a PhD student with Nigel Unwin at MRC, but worked out this structure when he was at EMBL as a young group leader, following in the footsteps of Dubochet. That was a crystal you can't see much because the contrast is lower, but they have a model here with 3 transmembrane helices and 13 or 14 bacteria chlorophyll molecules. So then that was how things were. People were beginning to make progress with EM, and then about 9 or 10 years ago now, a new electron detector came in, and up to this point, the best images you could take in electron microscopy were on photographic film. And so that meant you had to expose it, maybe 10 or 20 photographs, take it out of the microscope, develop it, fix it, dry it, scan it, and days would pass. But now we had these electronic detectors based on CMOS technology, the same thing that's in all the mobile phones or digital cameras. And they have 3 advantages compared to the old days, and this is what's produced a big step forward. One is the detective quantum efficiency for detecting the electrons hitting the image. Instead of being about 25 or 30% for film, it was 50, 60, 70, and now they're near 98 or 100%. And then the second advantage is these CMOS detectors, when you take an image, they take actually 40 or 100 or 1,000 frames per second. They take a little movie so that you can then, if the image is blurred due to charging or drifts of the stages, you can do motion correction and get a sharp image where you had a blurry image before. And then the third thing you can do is that because it's a movie, and because you're irradiating the sample and it's being destroyed as you take the images, you can do dose weighting. And each of these, the higher DQE, the motion correction, and the dose weighting, each of them produce about a factor of 3 or 4, so 33 to 100%, and then other factors. So you get a big improvement, and that has made almost a kind of mini revolution in electron microscopy. And then I'm just going to show you 2 or 3 structures that I think best reflect how things are. So, so this is the first one. So this is one of the many structures of different ribosomes that have been done using electron cryomicroscopy. And this one, these images, came from the work of Alexei Amunts, who was a postdoc at the LMB, with Venki Ramakrishnan, starting around about 2012 or 11, and trying to purify it and make crystals and do an X-ray crystal structure of these mitochondrial ribosomes. And the problem that Alexei had is that you don't get many ribosomes in mitochondria. You know, even if it's a beef heart mitochondria, you make the mitochondria and so on. And when you try to purify them, it's a mixture of the mitochondrial ribosomes with the cytoplasmic ribosomes. So it's only partly pure. It's a mixture of states, and then also the ribosomes can fall apart, large subunits, small subunits, and they have flexible states. But eventually he switched to doing electron microscopy and collaborated with Xiaochen Bai, who was in the group of Shor Sherez, and Shor's had written a new suite of software called RelyOn that allowed you to process images like this. And one of the things that Shor's had done earlier was to classify the images so that although you think they might look all the same, you can say, I think they're a mixture of states, that they're different. And in this case, they said, we think there are 6 states. And so the RelyOn software sorts them out. And it turns out that the 4 major states are indeed the mitochondrial ribosome that they were trying to find the structure of, but there's also one group that's the cytoplasmic ribosomes and another group that they weren't so sure about. And then if you look at these carefully, you can see one of them is just the large subunit of the ribosome, whereas another is a fully intact ribosome. And then the second thing that the RelyOn software from Shor's does, it allows you to focus the determination of the position and the orientation of each of those images of the ribosome onto the part you're interested in, for example, a large subunit. And then if there's any flexibility, and there is ratcheting in ribosomes, you then get, when you do the alignment with those orientations and positions, you get a sharp image of the bit you've aligned on. And by doing that, and then by doing another independent alignment, focusing on a different area of the structure, the small subunit, you can get a really nice map of the feature. And with that, they were able to get a complete atomic model of this 3 megadalton ribosome. And on the next slide, there is now some of the nice density in that map. So this is an RNA Watson-Crick double helix, 3 base pairs, and a top view here, and then an alpha helix and 2 beta sheets from a tiny part of that image of the whole ribosome. So you've got 3 megadaltons molecular weight, but many of the features are really well resolved like this. And their resolution here was 3 angstroms. These bases are 3.5 angstroms apart. So you can see that the resolution that's obtained is reflected in the density that you get. And then a second structure that I think shows the power of the method really well came from work that, from a project that Judy Hurst, who's in another MRC unit out at the Addenbrooke site, MRC mitochondrial biology, and she had been working on mitochondrial inner membrane complex 1, which is another big complex, 1 megadalton, 45 different proteins in that structure. But now it's in a membrane with lipids and has to be solubilized in detergent. And when you get it out and put it by the Dubochet method, you get an image like this. And you see images of the mitochondrial complex 1 in all different orientations. And with a little bit of work, Vinoth Kumar and Zhai-Pen-Ju, and so Vinoth was in the LMB, Zhai-Pen-Ju was in Judy Hurst's group in another MRC unit. They get this image at 5 angstrom resolution, similar to the myoglobin original structure in 1960. But you've now got 76 transmembrane helices rather than 7 in Bacteriodopsin, and then so 6 or 7 in myoglobin. At a higher contour level, you have these 8 iron-sulfur complexes. And the way that this enzyme works, mitochondrial complex 1, electron redox energy in the form of NADH is fed in at the top of this complex. It flows down these iron, jumping from iron sulfur to iron sulfur, and reduces a quinone in the membrane, which produces a structural change, and then 4 protons are pumped through the membrane, creating a membrane potential that can then be used by the ATPase to make ATP. So it's one of the major, probably, source of energy in the mitochondria that's in your body, makes muscle and all your body work together. And then with a little bit more work, well, at a lower contour level, you can see the lipid, and with a little bit more work, instead of 2014, it was 2016 then. Need to go. There's a bit of a delay somehow. Yeah, so in the end, they got a really 3.9, 4 angstrom resolution map in which they could build the amino acid sequence of all 45 different protein molecules. And in this diagram, now you've got atomic models for the iron-sulfur complexes. The blue part of the structure are 16 core protein subunits, and then the supernumerary ones are another 29. And the core subunits are the same ones that you find in homologs of complex 1 in bacterial equivalents. And Leo Sassanov, who was another group leader in the same MRC biology unit, had worked on a thermophilic complex 1 by X-ray crystallography a year or two earlier. And so that helped them initially in the building the main part of the model. But all the red subunits, 29 smaller, mostly smaller protein subunits, are the ones that are specific to mitochondria and were revealed by that structure. Now, of course, there are lots of groups doing related structures, different organisms, including Leo himself, who's now in Vienna. And this was a beef structure. Leo did a sheep structure, but there's lots of human structures and so on now. Okay, so one other structure I thought I'd show you, just to show you the breadth of the method, is this work from Miriam Koshai, who was a PhD student in Martinsried in Munich, with her supervisors Radisson Danev, and the department head is Wolfgang Baumeister. And Miriam said, I think we should try and look at hemoglobin by cryoEM. And prior to her work, most of us thought hemoglobin was a bit small, 60 kilodaltons. And of course, from the images, you have to find the particles and find their orientation. And if you look at this image, you can see them, but it's not so easy to see them. But the computer is better than the human at doing this. So these are her 2D classes. And with about 1 weekend's work, against the advice of her supervisors, she got this 3D model of hemoglobin with the heme group and the proximal and distal histidines, just like John Kendrew and Max had determined earlier by X-ray crystallography in myoglobin and hemoglobin. So those are 3 good examples. 2 of them, the complex 1 and the mitochondria, cannot be have their structure determined by any other method because of flexibility, impurity, and so on. Okay, so what about the impact of the cryoEM methodology for determining structures on a wider background? So here is the protein database. Now this is the parallel one. Cambridge Crystallographic Data Center in Cambridge is small molecules, but there's the protein data bank, which started out in the US, but now there's one at Hinxton, and one in Japan, and one in Rutgers. And they share every week all of the data. You can deposit anyway. There's now 185,000 structures. Most of them have been determined by X-ray crystallography. You can see there's been a kind of linear growth. It may have plateaued now. The EM structures are now in exponential growth. NMR was very productive in the early years, but for smaller structures, and peaked in 2007. And the comparison that we like to make, actually, is to try and look forward and say, what should we be buying more microscopes Should we be thinking of something else to do? Where is the future planning? So we like to plot this ratio of the number of structures determined per year, comparing X-ray crystallography to electron microscopy. And most structural biologists now are flexible and can do one or the other. But you can see 20 years ago, there was 1,000 or 2,000 times more structures. Very few done by EM, maybe 1 or 2 per year. And then these new microscopes came in with better vacuums and brighter sources. It was down to 100 or 200. And then the new detectors came in here, 2012. And since then, you've had, on a logarithmic scale, almost exactly exponential growth. And I looked earlier this week, and the current ratio is 2.4 for the beginning of 2022. And so it looks like there's no likelihood of it turning over quite soon, although it might do. And so we think mid-2024, it'll be about 50-50. And then this is all of them, all of the 3 different methods. And you can see all of them have periods during which the productivity of the method, once it caught on, you get this exponential growth, just like in computers and transistors per square millimeter and so on. So the X-ray was the first. You've got almost 100-fold improvement over that period that I told you when Max thought there were perhaps too many crystallographers, but it turned out that was before genome sequences came in, and now there's lots and lots of different topics. NMR grew even quicker, but over a shorter period, and then cryoEM is still in this exponential growth phase, and you don't know, it's very hard to keep going for factors of 100, 10,000, a million, but in transistor technology, they've managed to continue with Moore's law exponential growth for over 50 years. So they've had about 10 to 20 million-fold improvement in that. So we've only had something like a factor of 100 in each of these fields, so it will be very difficult to go forward. But one of the questions we asked was, what is it? Is it going to plateau, or is it going to carry on growing? So one of the comparisons we made, thanks to some help from Samir Velankar at the EBI in Hinxton, he provided us with the statistics from X-ray and cryoEM depositions from the MRC molecular biology lab. And of course, we had these detectors before anybody else. And so there's about a 6 or 7 year lead time here. And you can see here the crossover between half and half X-ray occurred about 3 or 4 years ago. And we're now here. So if you say the rest of the world will carry on in the same style, making their choices as they go along, you can see we're okay till about 2028. We don't need to worry too much about what to do after that. Okay, so now I'm going to show you, so that's a sort of general overview. Just going to show you a few new structures that are showing you how different people are using the methodology. And of course, there are now over 3,000 new structures being deposited every year by cryoEM. So there's no way you can go into too much detail about it. But I'll just show you a few. So one of the ones that we've worked on, and this is Xiaoxia Chen. Originally, we thought we just need 1 or 2 samples that will be standard samples. So this is human erythrocyte catalase. When you make a thin film and blot it by the Dubochet method, instead of getting lots of nice particles, which would be down here, they tend to form these ribbons. And it took quite a lot of effort to get rid of these ribbons so that you could get views of the molecules in all different directions. And so this was after adding 1 detergent, and you can see that the ribbons have gone, but the orientation has been retained for most of them. And when it's been blotted to a very thin film, the catalase molecules are squeezed to a very thin film to be about 100 angstroms thick. And so you've gone from 1 view of the molecules to 2 views of the molecules. But with a different detergent, this was Chapso, you get them in all views. But of course, the reason that you're getting this is that the molecules are interacting with the air-water interface. And so you have to increase the concentration, and then you get it in all different orientations, and then it's relatively easy to go with a few hundred micrographs. You get 1 1 mg per mil, 10 mg per mil, 20 mg per mil, and you can get, instead of one orientation, you can get lots of different orientations. Then you get the structure. This is the overall structure. This is a molecular model of it. And here are the heme group and the NADPH here. And just like in mitochondrial complex 1, NADPH donates an electron that reduces the heme and allows it to carry on in this hydrogen peroxide removal so that you don't get damage to your DNA. And then that's a higher resolution picture of the two ligands and an alpha helix and a beta sheet from two different directions. And then another structure was done by Vinoth Kumar, who worked on mitochondrial complex 1 with Judy Hurst, and Giuseppe Canoni, and a group from India who had been working on this phenylacetic acid degrading enzyme. It's a simple enzyme, but they collected 4 data sets, one without ligands, and then one with ligand 1, ligand 2, and then both of the ligands. And the ligands are NADPH and octanyl-CoA. So you can see then, by collecting different data sets and subtracting the two, you can find out where the ligand is, what its structure is, and how it's interacting with parts of the protein. And then that opens the way for the idea of using cryoEM to do drug design, just like the X-ray crystallographic community have been doing for 30 years with drug design, and which has been used in that Paxlovid X-ray structure that I showed you. So this is from a paper, one of the first papers published by pharmaceutical drug companies, and this is Astex, which Tom Blundell was involved with starting a while ago, Haran Jyoti, and then Pamela Williams was an X-ray crystallography, but got involved with cryoEM, and they took a test structure, beta-galactosidase, that's the LAC-Z gene from E. coli, the first operon, and did a number of, many more than this. In this paper, they have about 60 data sets, but here are 3 ligands that are inhibitors of the beta-galactosidase, and this is the way that you do drug design against, for example, a virus target to make antivirals that would prevent infection. But this was a test structure, but here is one that's a genuine anti-cancer target, a particular pyruvate dehydrase. So many drug companies now, probably not quite as many as have been doing X-ray crystallography, but getting deeply involved in it. And you can then also use the cryoEM to look at the structures of things that were previously extremely difficult to work on. So here is one structure of a Gproteincoupled receptor complex. So it's the adenosine A2A receptor, which one of the companies in Cambridge that have been involved in getting into cryoEM, Heptares, have developed a drug that assists in certain types of cancer. So here is the receptor itself, and this is the alpha beta gamma G protein complex bound to it. This is from work in Chris Tate's group done by Javier Garcia Nafria, who was a Spanish postdoc who came and started taking images like this, which were similar to the ones I showed you, but involves a faceplate, and these are the 2D classes. Here is the density map, and here is the interpretation of it in terms of atomic model. And this would be the ligand that would be bound in an adenosine analog, activating this G protein coupled receptor. And if you were designing a drug to be an agonist in this case, you would know how to go about modifying it by looking at that structure. And then one last one is another structure from Javier Garcia Nafria in Chris Tate's group, which is 5-hydroxytryptamine, serotonin, GPCR, again with an inhibitor bound or an activator, donitriptan bound. Again, similar images, similar 2D structures, a nice high-resolution structure, alpha helical parts of the structure. So people are using these methods then to do. So it's working now, and it's being productive, and it's growing in power, and we're fairly confident that it'll grow for a while, provided there are enough people and enough investment in the hardware. But I thought I'd just finish with 2 more slides because of the topical nature of the coronavirus. So one of them is a picture of the whole SARS-2 coronavirus, which is only from 2 years ago. On the left are schematic diagrams taken from Wikipedia. This is a artistic diagram. The artists do it really well. But on the right are real electron cryomicroscope tomography images of 3 different virus particles done by the group of John Briggs, who was at MRC and is now in Martinsrud in Germany. And you can see the spike proteins. This is the protein that binds to the receptor on the cell that it's infecting, sticking out from the surface of the virus. When it's, this is the sort of, as the virus enters your body, it's like this, and then it changes its structure and fuses with, and then puts the RNA into the structure. So that's electron cryotomography. But then there are, and I mentioned before, there are several, 4,000 or 5,000 protein data management depositions in the last year to do with these 3 drug targets in the coronavirus. And so this last slide is just one of these many, many papers from Yao Kong's group in Shanghai, where they've determined the structure of the spike protein trimer on its own and bound to the receptor binding domain, which is part of this angiotensin converting enzyme that's on the surface of the cell that it binds to. And using structures like this, the people who are trying to design inhibitors to prevent that interaction are making good progress, just like they're doing with the RNA polymerase and the protease. So then I'd like to finish by just looking ahead a little bit. So where do we think things are going? Well, we don't think structural biology is running out of steam. So the 3 main directions are what's going to happen with electron microscopy, X-ray crystallography, and then what can you do with computing, which perhaps bypasses the experimental needs. And with electron microscopy, we are hoping that you can keep going in this exponential growth phase and avoid plateauing it by making the microscopes less expensive so that more people who would like to use it for their favorite topic don't have to queue up for access to microscopes in another department where they're busy doing something else, or go to a national center. So we've got this plan, hopefully, to help to develop less expensive microscopes that work just as well. And then other people are keen on extracting images from tomograms. That means without purifying the protein molecules, so that you can search the tomograms or images of unpurified or partially purified preparations, and then get the structure without having to do a lot of the preliminary biochemical work. So there's a lot of potential there, and people will think of new ways of approaching it, provided they can get access and try all their bright ideas out. And then all the synchrotrons are in the process of upgrading to make them brighter, and that will have the effect that you can either collect data quicker, higher throughput crystallographic beamlines, or it makes it particularly good for X-ray microscopy, X-ray tomography. And there are a number of groups now that are doing not atomic resolution structures, but sort of nanometer resolution structures. And for example, there's a group, Peter Lee at University College in London, where they've looked at the entire lung of a normal dead person and a person that had been infected with coronavirus, and you can compare and look at how all the different blood vessels and so on. That's comes only from brighter synchrotron upgrades, for example at ESRF, but it'll be coming in the UK as well. And then also, sometimes you don't want to freeze your samples, and then there are now a number of free electron lasers where you can get femtosecond pulses and then get all the diffraction without worrying too much about radiation damage, and that's particularly powerful at room temperature. So there is great potential, particularly in electron microscopy, particularly electric crystallography, and then for many years, I mean, even in 1985, the people who did computational biology, where you try to compute the structure or compute how a ligand binds to a known protein structure, were saying they could do it, but the energy functions have just not been very accurate. But they're getting more accurate all the time, and we're hoping that eventually you'll be able to do protein folding, drug design, with a much higher probability and hit rate than you can at the moment. And of course, there's AlphaFold, and there's differences of opinion as to how powerful that was. Sometimes it works really well, sometimes not. So I'll just finish then by acknowledging, I mean, it's been great fun for me for 50 years doing structural biology, but these are some of the people that I've had great fortune to work with, particularly Nigel, who's up at the back here. And without Nigel, I wouldn't have been ever getting involved in electron microscopy. And then we spent years and years and years trying to get to higher resolution bacteriodopsin. And Bob Glaser and Ken Downing in Berkeley, and then Fritz Semlin and Eric Beckman in Berlin, and Jean Le Paul, who was in the group of Dubochet at EMBL in Heidelberg. And then Joyce, Tom, and David were here in Cambridge with me. That was a big, long effort there. And then we also spent a long time doing this. We were one of the groups doing this detector development, which really did help a lot. Also, there was other groups in America, but Greg and Wazi here, and then Renato and Nicola, who are Italian CMOS chip designers, were greatly involved in the detectors we were helping to develop. And then Xiaoxia, who is the manager of our EM facility, but has also been involved in a lot of these projects, including that catalase project. And then we didn't get involved in single particle EM till about 1995, when we stopped doing electron crystallography and started to doing electron microscopy on single particles. And again, that took a long time, so 1995 to 2012. But these were some of the people who were in the lab. Sriram and Jacqueline are now in Vancouver in Canada. Nico's at UMass in Massachusetts. John Rubinstein, Toronto. Peter Rosedal at Crick in London. And Vinoth in Bangalore, leading the Indian cryoEM facility. And then Chris is a group leader at the LMB, and a couple of the students from Chris's group are here this evening. So that made a big difference. Then in the talk, I also showed work by other people. And so I just wanted to mention them. So Dubochet's Adenovirus and all his plunge freezing, really, his work is the thing that opened up the field. And we even had Linda Amos and I in the lab. We had a student that was working on one of our projects on tubulin, and we sent him to an EMBO course on the early stages of cryoEM. And when he came back, he resigned from our group and said, I'm not going to do your work anymore. I'm going to switch to doing electron cryomicroscopy. And he switched to working with two other group leaders in the LMB. And that was one of the earliest projects here in Cambridge using the Dubochet method at that point. And then I've mentioned the two projects that were in the LMB, one by Miriam Kashai in Martinsried, and then he said 3 other Cambridge-based or 5 other Cambridge-based projects, and then some highlights from the SARS-CoV-2, where none of us were allowed to go to work unless we worked on SARS-CoV-2 during the period of about 2 or 3 months, about a year and a half ago, something like that. So I think that's hopefully given you a bit of an idea about how cryoEM has made an impact on finding out more about the structure of the biological molecules in life, and is complementing earlier pioneering work by the X-ray crystallographers. And we're all looking forward to continuing progress in the field and continuing productivity. So thank you very much. Yeah.